Good afternoon, good evening, and good early morning to you, wherever you might be calling in from. We appreciate your presence today. I am Rob Burgess, and I head up business development for Sino Biological. And it is my pleasure and honor to welcome everyone once again to the next installment in our series called Biotalk Tuesdays, where we hear about cutting edge technologies, products, services, therapeutics, and diagnostics offerings from around the world. We have a very special speaker today who's going to tell us about nanotechnology and its relation to rare antibodies and T cells. But before I introduce our speaker, I just wanted to mention one very brief housekeeping issue. I would like to ask everyone to withhold their questions verbally from the speaker during the talk, but please enter those into the chat box if you would. And at the end of the seminar and lecture, I will run through the chat box and get to as many people's questions as I can. So I will ask them verbally on your behalf to our speaker. So please do. And also while you're at it, just type into the chat box, say hello, and tell us where you're from. We're always interested in where everybody's calling in from. So with that today, it is my honor and pleasure to introduce to you our speaker for this installment of Biotalk Tuesday. It is Dr. Dino DiCarlo. And Dr. DiCarlo is the Armand and Elena Harapetian Professor of Bioengineering at the University of California, Los Angeles. He serves also in academic leadership roles as the Vice Chair of the Bioengineering Department and the Deputy Director of a National Science Foundation Engineering Research Center. He is an author on more than 170 peer-reviewed articles and an inventor on more than 70 issued patents in the world, in the United States, as well as worldwide. He has received numerous awards, including the Presidential Early Career Award for Scientists and Engineers. And this is the highest honor that's bestowed upon young scientists and engineers within the United States. He is co-founder and director also at Cytometry, an 80-plus person company commercializing an FDA-cleared sepsis diagnostic test for rapid triage. And then finally, he is scientific advisor to several life sciences companies, including Q Health, which developed the first at-home molecular diagnostics test to be cleared by the FDA. And with that, the title of Dr. DiCarlo's talk is Next Generation Nanovial Platforms for Function-First Discovery of Rare Antibodies and T Cell Receptors. Dr. DiCarlo, welcome. It's a pleasure to have you. And I believe Max will now turn over the screen sharing to you.
Great. Thank you so much, Rob, for the invitation to speak and the kind introduction. Thanks, everyone, for attending this talk. And good morning, good evening, good afternoon to you all. I see that we're from all around the world, which is great. This is the new technology allows this, and it's my pleasure to share some of the work that we're doing, and hopefully help you with research that you're conducting in the field and learn something new today. Okay. Are you seeing my screen?
Yeah, it looks great.
Okay. Well, so what I want to talk about today is a new platform that we've been developing, which we call a nanovial platform, which allows us to look at the functions of individual cells. And so this platform is in essence, a test tube for a single cell. And we can use millions of these individual test tubes to perform assays on millions of individual cells. In particular, we're interested in looking at things that they secrete, which relates directly to their function, in particular functions like ability to secrete antibodies and for antibody discovery. So these, for example, like plasma cells or hybridomas, and the ability of T cells to secrete cytokines or other effector molecules, which are important for their function in targeting, for example, cancer cells. So those are two areas that I'm going to talk more about. Are my slides advancing? Just want to confirm.
Yeah,
Yes,
yeah,
they are.
they sure are. No
Okay.
problem at all.
Great. So how I like to frame our interest in developing this type of technology is in what I refer to as the three pillars of medicine: molecular therapies, gene therapies, and cellular therapies. In my lab in particular, we're interested in developing tools to enable development of therapies across these three pillars. Molecular therapies, this is something that the pharma industry has focused on for the last several decades, particularly on small molecules initially, but more recently on biologics, and in particular monoclonal antibodies, with now 5 out of 10 top blockbuster drugs being monoclonal antibodies. So this is an area that discovery of new monoclonal antibodies, in particular using plasma cells that are secreting antibodies, that can have a big impact on the field and improve the types of therapies that we can bring to market and to patients. Gene therapies is the second big area, and we've seen a lot of excitement and new clearances, new approvals from regulatory agencies of gene therapies, including, I guess, in the UK, a recent CRISPR-based approach for treating a genetic disorder. One of the things that is a barrier for these therapies, though, has been producing GMP grade virus. And so that's an area where if we can select cells that release more virus and start to understand what causes a cell to be a good producer of virus, that could kind of unlock new capabilities, reduce bottlenecks in producing gene therapies. So we're looking at tools that could help do that as well. And finally, cell therapies, super exciting area. Unlike molecular therapies, where a molecule is interacting with another molecule and blocking or activating its function, cell therapies are like computers in some ways, right? That they have not just the ability to block functions or activate functions, but they have the ability to sense their environment, make some computational decision, and then actuate or create some therapeutic effect in response to the environment. And this is what everyone's excited about. And we've seen new approvals over the last 5 years or so, especially in the chimeric antigen receptor T cell therapies. And so these are therapies that target antigens on tumor cells and then elicit effector function in the T cells, but also those cells are living drugs, and that they also are proliferating and responding to their environment, and even bringing other cell types into target tumors. So very exciting direction. When we want to discover new chimeric antigen receptors or new T cell receptors, I'm going to talk particularly about T cell receptors because they're important in targeting especially intracellular antigens. That is a difficult process to find those rare, high affinity cell receptors that also, or tuned affinity receptors that also lead to a particular level of secretion of cytokines or other effector molecules for cell killing. So that's a process that we want to enable. Besides these chimeric antigen receptor T cells and engineered TCR therapies, we have also new therapies where cells are used to secrete drugs. So they're implanted, maybe the most well-known is secretion of insulin from iPS-derived islet cells, but you also have now excitement in being able to even secrete antibody drugs with implanted cells. And so this is a direction where we think our tools could be helpful as well. So as I mentioned, we're interested in function of cells and screening based off of function for enabling all of these areas, and antibody discovery and cell therapy discovery, engineered cell therapies, even biomanufacturing of viruses, extracellular vesicles, other biologics. In all these cases, what cells are secreting are the key aspect of the behavior of the cell that we want to select based off of. And in fact, there's over 3,000 secreted proteins that are predicted to be secreted in the human genome. And that's a similar amount to the number of surface markers on cells. We have a lot of tools to sort cells based on their surface markers, but very few to sort and isolate cells based on their secretion. So that's really important for us. And these secretions play an outsized role in the function of cells, right? So we know that you think about it, the way I like to think about this is a cell contacting another cell and communicating that way is like me whispering to my neighbor, but a cell secreting is like me taking the megaphone and shouting out so that everyone can hear. So that has an outsized effect on the biology in vivo. But the challenge is, right, that researchers really don't have great ways to sort cells based on these important secreted factors. And we think that the technology to do that will be really important. And why don't we have great ways to sort cells based on secretions, in particular single cells? Is the vessels that we use to perform biology really aren't matched to the scale of the biology. So we use things like bioreactors, well plates, even at the smallest 1536 well plates, or we're still at length scales that are hundreds of times larger than cells, and even that means volume scales that are tens of thousands of times larger than cells. And why is that a problem? Well, if a cell is releasing few numbers of molecules into its environment, that gets diluted out in this huge volume, right? And so if you can concentrate all of that information into a small volume around the cell, so that allows you to detect it a lot more easily. And that's where tools like microfluidic tools have come into play. I show on the bottom here an image of a droplet generator. And I've worked in the microfluidics field for a long time. And this is a great approach to shrink the volumes in which you can perform assays. But one of the challenges in that field has been that I've seen through commercializing microfluidic devices is that often you have a very simple chip, but you have a very complex instrument around the chip. And that is a barrier for others to adopt this technology. And as we have folks from around the world attending this webinar, not everyone has instruments to perform microfluidic assays or the expertise to do that. So our thought was, can we leverage the infrastructure of life science research, things like flow cytometers that already are existing in research labs, and to some extent single cell sequencing technologies that are already present in labs like the 10x Chromium system, and have our assays, have our nanovial technology, which I'm going to show here, lab on a particle based approach that we call it, have that be compatible with all that infrastructure. And so that all of your readout, all of your asset analysis is done using current infrastructure that's already in place, but a new reagent or a new particle that is easily adopted. So that was the goal. And so what are these nanovials? Well, they are hydrogel particles that have cavities that are smaller than a nanoliter. So I'd like to show this 3D printed version of a nanovial here in my hand. It even matches the color on the screen. But basically, this is obviously not to scale. A single cell fits into this cavity. But this is a transparent hydrogel particle that has a little cavity in it, like a little bowl. And we can put millions of these particles into a tube, and we can load cells into them. So here's just a little video that shows some of these nanovials as they're settling in a well. We can make them extremely uniform. In this case, where we had particles that were a bit larger, about 80 microns in diameter, with cavities about 50 microns in diameter, but we make particles all the way from 35 microns in diameter to 100 microns in diameter. The sweet spot where a lot of the work that I'm going to show is based off of our particles about 35 to 50 microns in diameter. So what do these particles do? Well, they create compartments, right? As I mentioned, they're kind of like test tubes for single cells. So the cells can fit into these compartments, and then they also are a solid substrate on the hydrogel to perform reactions. So we can include antibodies and other things, or antigens, as I'll talk more about. And because they're small enough, they have their ability to analyze and sort them using standard cytometers. So this shows you a little bit more detail in a cartoon of what's happening on the nanovial. We have some usually antigens or other things that will interact with the cell, or could be an antibody that allows the cell to bind specifically in that cavity and stay there. We also include antibodies to bind to things that the cell is secreting. So imagine now you have a cell in the small cavity. As it's secreting, it's accumulating in that cavity. The cavity protects it from flows that would wash away those secretions, and those secretions get captured in the cavity. So what does this look like? It looks something like this, these transparent hydrogel nanovials. You have cells that are labeled in pink here, and all the secreted, in this case, IgG, these are CHO cells that are secreting IgG, is being captured in the cavity. And you can see that fluorescence as we label it, that secretion afterwards with an antibody that is fluorescently labeled. So just like you might perform an ELIS spot, and I'll talk a little bit more about that here later, here we kind of have a sortable ELIS spot that all of the information that the cell is secreting gets captured locally, is associated with that same cell, and that can then be sorted with standard flow cytometers. Here I show a picture of the Sony SH800 that we often use in our experiments. So what does a workflow look like using these nanovials to perform these single cell assays? So the first step is you load cells into the nanovials, and that's just pipetting cells and nanovials together in order to basically have them have enough contact and kind of mixing so that they can get into the cavities. Once they're in the cavities, they can bind specifically, as I mentioned. The nanovials also have a secretion capture antibody. So when you incubate them for a period of time, as they secrete, that secretion is captured, and so it's associated with the cell. You can then wash away excess secretions and things, and then stain the bound secretions using an antibody that is fluorescently labeled, or another antibody that is tagged with an oligonucleotide. So why is that important? Is that if you want to link information about the DNA or RNA or gene expression of the cell with what it was secreting, you can now do that as well by putting this whole nanovial, for example, into a single cell sequencing workflow. So after you label the cell and the nanovial and the associated cell, you could actually also label surface markers on the cell. You can then sort that nanovial cell complex, regrow cells so that this actually protects the cell from shear stresses and increases the viability of the cells. You could look at productivity of those cells for secretion of interest, like an antibody, or you could go downstream and perform sequencing. So in this case, single cell sequencing in order to extract information on the transcriptome, or what I'm going to talk about is sequencing the heavy and light chain for IgG recovery, and sequencing the alpha and beta chain for TCR. So these VDJ sequences. So how do we make these nanovials in the first place? We actually use microfluidics in the process of making nanovials. We have two streams, a PEG polyethylene glycol stream that has cross-linkable moieties and biotin, in this case norbornene. We also have another sugar or gelatin or some other large polymer molecule that co-flows with this PEG. When PEG and dextran are mixed together, they phase separate. It forms what's called an aqueous two-phase system. So you get a droplet that's dextran rich and another region that's PEG rich, and it actually forms this concentric shape that we refer to as the nanovial shape. So the kind of in cross section, kind of a crescent shape. We can then cross-link the PEG with UV exposure, and then wash away the dextran that was in the center, and that forms these cavity-shaped particles. As I showed previously, we can create a suspension of these, and that then used by when we want to perform an experiment, or we ship it to others to perform experiments. We refer to this as microfluidics 2.0, and that we're kind of getting rid of the initial microfluidics. That's all done by by a manufacturing group, and then the actual users do microfluidics without having to deal with all sorts of microfluidic chips and all of that. So we're just starting to kind of get a lot of this work out there, starting really last year and building into this year. Including work that's in Nature Communications this year, ACS Nano last year, and Nature Nanotechnology this year, across many different application spaces, from what we call secretion encoded single cell sequencing, or seq-seq, where we're linking secretions with the transcriptomes of individual cells to, as I'm going to talk more about today, work that is focused on looking at antibody secreting cells, based off of this paper and some other work from our collaborators, as well as defining T cell receptor repertoires that are specific, for example, to cancer antigens. So, so I want to start by talking about why do we want to apply this technology to antibody discovery. There's definitely challenges with current processes for antibody discovery, whether it's hybridoma, direct B cell antigen baiting, phage display, et cetera. And so, what we're focused on is looking at isolating antibody secreting cells like plasma cells that have affinity matured antibodies that we know are secreted in a way that that they're developable, that they're not going to aggregate, and that they have matched heavy and light chains. And so the process really here is that you can perform this whole workflow of going from immunization, plasma cell isolation, looking for antigen-specific secreted antibodies, to sorting and sequencing for hundreds of thousands to millions of cells in parallel using these nanovials. Because we're looking at plasma cell, that allows you potentially to have increased affinity antibodies, larger diversity of candidates. Because we're using flow cytometry, that gives us a high dynamic range of our fluorescence intensity, so we can look at affinity effects. And then that also allows us to quantitatively assess that in the sorting process. So we can see which sequences of antibodies are associated with which kind of fluorescent signal, and we can rank when we are re-expressing to characterize them. As I mentioned, the nanovials protect cells from shear stress, so that enhances viability, especially if this is helpful if you're looking at hybridoma cells that you want to regrow. And then, as I mentioned as well, compatible with standard flow cytometers. So that's great from a perspective of just being able to adopt and use the technology. So I'm going to show one workflow that we've done, we've internally performed for antibody discovery, kind of just to show the proof of concept of antibody discovery. And then a few slides from our collaborators that have done campaigns for antibody discovery on plasma cells. So the first step, cell loading. In this case, we used an CD45 antibody to bind the cells of interest. We had an anti-IgG antibody on the nanovials. In some cases, we can encapsulate in an oil phase, but we mostly don't do that nowadays because it's not necessary to get specific signal. So we're looking at secretions that accumulate on the nanovial. And then we label with a fluorescent antigen, and then we sort to individual cells for doing single cell PCR. So in the first kind of proof of concept, we looked at hybridoma, the HiHel-5, or hybridoma that produce an antibody against hen egg lysozyme, or HEL. We mixed in or spiked in HiHel-5 into a background hybridoma line 9E10 that did not produce an antibody against HEL. So you can see here, when you have a HiHel-5 antibody that has no cell tracker on it, we can see that there is this high fluorescent signal for associated with the antigen, antibodies that bind the antigen. For the 9E10, we don't see that, and we put a cell tracker to also just confirm and allow us to calculate our efficiency. So this is what some of the flow plots look like when you operate with nanovials. So here are forward scatter and side scatter. We gate on this population. And then we gated on the population that had high signal in the 647 channel. So, and then we gate this 0.18% of the population with very high signal, excuse me, in that channel. And what we found is that from a pre-sort spike, we spiked in about 4% of the HiHel-5s, and then we enriched that population to 90% after we sort out this population. So after we sort out this 0.18% population, we have a 90% purity of the HiHel-5 hybridomas. And you can see the kind of what they look like post-sort when you have cells that are secreting on the nanovials. We could then perform single cell RT-PCR, and this is just showing heavy chain being amplified. in a well. But you could also, as I'll talk about, we can go into a downstream single cell droplet-based sequencing workflow. And we could also clonally expand the hybridomas that were sorted. This is day 0, the hybridoma on the nanovial. At day 3, it kind of expands out, covers the entire nanovial. As you go even further days, then it will just, cells will expand even further beyond the nanovial and fill the entire well. Okay, so so that was kind of the proof of concept that this is possible to do with hybridoma cells. And then I want to show a few slides from our collaborators at Partilian Bioscience, so Joe Dorette, and Dilip and Jenny, who've used this technology at Alloy to perform a plasma cell workflow. And so they start with their mice, they immunize, isolate from bone marrow plasma cells. They load using an anti-CD138 antibody, filter out any background cells, incubate to allow secretion. In this case, the workflow is a bit different in that the antigen is biotinylated, put on the nanovial, and secreted IgG is bound. And then an anti-IgG fluorescent antibody is used to detect the antigen-specific secretion. Those cells are then sorted that are antigen-specific into well plates, and then amplification of the heavy and light chain is performed, re-expression is then performed, and then functional assays of the discovered antibodies. Here's what that same workflow here shown on the top. Here's what the flow plots look like. Cells on nanovials are gated, CD138 positive cells are gated, and then cells that have signaled from antigen-specific IgG are gated and sorted. Those cells were then sequenced. Here's kind of showing 130 plasma cells that were isolated that comprised 53 clonotypes, 21 heavy chain, and 15 light chain families. So some pretty good diversity. And this was sorted with this FAX-ARIA 3. When those were re-expressed, what was found is, they found these picomolar, sub-10 picomolar binders, many sub-10 nanomolar binders that bound protein, but also then bound also antigen on cells. In this case, this was an antigen that was a cell surface protein. So very exciting that they could find these very high affinity antibodies using the plasma cell secretion workflow on nanovials. And this is just showing some of the kind of similarities between the different antibodies that were found. So some diversity, some that bind at similar epitopes, but pretty exciting discovery campaign here. Okay, so we're really happy about how this nanovial technology can be applied to antibodies. And I want to now transition to how we've been applying this with our collaborators to also functionally screen T cells. In essence, this is a similar workflow as for antibodies, right? We're looking at, instead of IgG that is specific to a particular antigen, we're looking at T cell receptors that are specific to peptide MHC molecules on the nanovials. So this is collaborative work that was led by Felice, who graduated from my lab just recently in my lab, and then Mao in Owen Whitty's lab, and we also collaborate with Lily Yang's lab here at UCLA. So why are we interested in TCR discovery? So as mentioned at the beginning of the talk, we're very interested in finding new antigen-specific TCRs that could be useful to engineer into other cells as a way to have a new cancer therapeutic. And in particular, this could be neoantigen, looking at cells that target neoantigens, that could be targeting viral proteins associated with cancer. It could be targeting splice variants or other differences between normal cells and tumor cells. But often these TCRs are extremely rare. So they're in a diversity of 10 to the 20 potential TCRs. The rarity of these reactive T cells against cancer-specific antigens might be 1 in billions of T cells, hundreds of millions to billions of T cells. So that's a problem, because then if we have low accuracy with our current approaches using affinity or activation biomarker-based discovery, that leads to lots of extra work where we have TCR sequences that aren't really functional, right? So we're getting, for example, and work that was done by the Whitty lab previously, they looked at 14 distinct healthy donors. They did, through their affinity and activation biomarker-based processes, they found about 400 different TCRs, but the reality is only 8 of those were actually functional. So like 2% of the TCRs that were recovered that were supposed to be functional were actually functional. So can we improve that so that we're not re-expressing a bunch of garbage TCRs that actually don't work? So reducing the false positive rate in discovery, I think, is one challenge. So improving the accuracy. And so one way to do that is not just look at affinity or activation, but to look at combinations, look at function. As I meant, as the title of my talk is, function first. Can we look at the T cell function as a way to at the first screening step, as a way to narrow down, make sure that we are really only finding the T cells that are really active, and that their TCRs that we then sequence are real TCRs that are going to be functional. And so looking at binding, but then secretion is a great way to do that. So bottleneck 2 is that there's a growing evidence that the types of cytokines, or the amounts of different cytokines that T cells secrete, is helpful in understanding if the patients that are given an engineered cell therapy, like a CAR T cell therapy, will respond, right? And so there may be something about the kind of functionality, or what was referred to as polyfunctionality, that is important for these cells, the engineered cells, to be useful in vivo and be responding. So if we can select out and we can sort and understand this polyfunctionality, that can help us manufacture perhaps better cell therapies as well. So one of the challenges for both of these is being able to screen and then select out cells based on function. And so that really can help us improve the discovery process, as I mentioned, as well as manufacturing of cell therapies potentially. So what are the kind of current technologies to look at cell secretion? The kind of bread and butter technology is ELISpot. For single cells, you put cells down on a plate. You allow them to secrete. As long as you don't mix the plate, the secretions kind of accumulate locally, form a bloom around the cell. You can then label the secretions with a second antibody, and then understand how many cells we're secreting. This is great. It has some advantages in terms of kind of simplicity, but you are not getting other information from those cells. You cannot recover those cells based off of the function. So that's critical. That doesn't allow you to do the T cell discovery workflows that we want to do. Another recent really great technology from Isoplexis, now part of Bruker, uses this kind of microfluidic chip to look at multiplexing in many secretions. And so it's kind of like ELISpot, in that you put cells on a surface, they secrete, you capture the secretions, you can label them, but now it multiplexes that by compartmentalizing cells into individual containers. Some of the challenges here, still, you cannot recover these cells based on function. So that kind of mitigates the use for TCR discovery, or prevents it from being used for that. And then there's some limited throughput. And the third is this Miltenyi cytokine catch assay, where you have a bispecific antibody to bind CD45 and maybe a cytokine of interest. And so you capture on the surface of the cell the secretions. And so this allows you to recover cells based off of secretions. But in our hands, we've seen crosstalk between cells because there's nothing preventing those secretions from being convected away. And it really can't be applied to all the types of things that we've been looking at, for example, antibody secreting cells. So, and I think the final point is none of these technologies enable you to also characterize that affinity interaction, or to enrich cells based on binding to peptide MHC in the case of T cells, that allows you to both use the nanovial in our case as a way to stimulate the cell, bind to stimulate the cell, and also captures the secretion. So it has this multi-function functional purpose that you don't see in these other technologies. So how are we doing this T cell work? Well, Well, yeah, it's very similar to the other work that we coat the nanovials with some reagents. So we coat them first with peptide MHC monomers that are biotinylated. That allows us to have this beautiful lawn of peptide MHC monomers that T cells can interact with. In some ways, that creates a very high avidity compared to like tetramer-based approaches because you have this huge polymer surface covered with monomers. We then also have capture antibodies. In this case, I'm showing interferon gamma. And then finally, we have an oligonucleotide barcode that barcodes this particular nanovial that had a particular antigen, peptide MHC monomer, so that I can know later on when I sort that cell out and I sequence its TCR, which monomer did it bind to? Was it, in this case, the CMV1 specific monomer or the Epstein, so in this case, cytomegalovirus or Epstein-Barr virus specific monomer. And so what we've done, and I'll show later on, is we've multiplexed this up to like 10 different peptide MHCs that we can do simultaneously so that one batch of cells, you're screening across many different target antigens. So after we modify the nanovials, we load cells, we allow them to secrete for about 3 hours to accumulate cytokines, interferon gamma in this case. We then label the secreted interferon gamma, and we label cell surface markers. We sort out the cells, the CD3 positive, CD8 positive cells, so the cytotoxic T cells that are also secreting interferon gamma when they engage with this peptide MHC surface. So that means that there's a specific reaction that's occurring, that they become activated from this kind of artificial antigen presenting cell, and then they specifically secrete cytokines upon that activation. We can then sort out the cells based off of that secretion and then perform the downstream single cell sequencing using, for example, 10x chromium VDJ sequencing, or we can look at proliferation of those cells. So the first thing we wanted to test is, do we see specific binding of T cells that have a particular engineered TCR, in this case 1G4 to the NY-ESO1 peptides. And so we looked at binding as a function of including, having the specific TCR, the cognate TCR, and you see increased binding as the peptide MHC concentration increases. And then we sort out, when we sort out the cells that are binding, we see that they also mostly express this NGFR signal. The NGFR signal is in the engineered TCR construct. Finally, we wanted to make sure that there was selective activation of cells due to the peptide MHC on the nanovials. And so the 1G4 cells that interact with peptide MHC, we see this beautiful interferon gamma positive secretion signal, while on the 1G4 cells that interacted just with a CD45 nanovial that still could capture them, we don't see, this is in the red here, we don't see the strong interferon gamma secretion signal. Just briefly, cells that are isolated with this process, we could sort them and regrow them, and the cells that were specific that had the NGFR signal were the ones that proliferated and regrew. Okay, so now that we kind of showed the proof of concept of this type of TCR T cell assay where we could isolate based on antigen-specific TCRs, look at their function, their secretion, and then sort them, we wanted to kind of show how we could perform assays with this on PBM, starting with PBMCs. And what you see here is enrichment, with enrichment, without enrichment. So there's two workflows that we've done. One is we give a pool of peptides to PBMCs, peripheral blood mononuclear cells, allow antigen presenting cells to present antigen to cognate T cells. Those cells get enriched, and then we can run an assay on the enriched cells. And then without enrichment is directly from PBMCs, and that's what this is showing here. When we go directly from PBMCs, we can gate the CD3, CD8 positive cells that are on nanovials, and then we see that some fraction of those become that secrete interferon gamma. We can then sort out that fraction that's in secreting interferon gamma, and we see from starting at 10 million cells, we're seeing about 400 cells that are CD3 positive, CD8 positive, bound to nanovials, and then secrete interferon gamma. When we use a tetramer-based approach to look at these same cells, in this case, it was a CMV specific cells, we only found about 163 cells that were beyond the negative gate, starting with the 10 million cells. So it looks like we're enriching more cells, but the question is, are these cells actually cells that are true TCRs? As I mentioned before, we don't want false positives. So we then went through a full workflow where we then sequenced the TCRs and then re-expressed them. And so let me show you what that workflow revealed. And in this workflow, again, as I mentioned, we barcoded the different peptides on the nanovials, in this case, 3, CMV1, CMV2, and EBV, with specific feature barcodes. We compared then the nanovial-based assay with CMV-specific tetramer staining, and then that's an activation-based, affinity-based, and then an activation-based was based on CD137 staining of the activated cells. So maybe the key comparison here is the nanovials is an affinity plus secretion-based sorting approach. So we're looking at binding, specific binding, and then activation of secretion versus an affinity-based or an activation-based protocol. This is kind of some of the flow plots from this workflow. You have nanovials being gated, CD3, CD8 positive cells, and then interferon gamma positive cells. And then this is, after the sorts, what some of these nanovials look like. And so we activate them, collect the secretions, and then sort these positive cells. We then went into the 10x chromium for VDJ sequencing of those selected cells. So just briefly, this follows kind of the standard process if you would just put cells into these droplets. But here you can see that there's a nanovial that has a cell and a nanovial. There's the gel bead, and here's the droplet. So all that fits into this droplet. When you lyse the cells, you see the lysate kind of accumulates in those droplet. And so this is very compatible. We see quite compatible with this workflow, and that's allowing us to get TCR sequences back. So here's some of the statistics of the recovery after the 10x workflow. For tetramer-based approach, we found about 800 cells. And then for the nanovial-based approach, 6,000 or so, and 6,000 for a CD137-based approach. And then, there's some loss through the single cell sequencing workflow. But, the number of cells that had V to J spanning pairs were about the same or a bit larger for the nanovial-based process. So the nanovials did not interfere with the sequence recovery. We then looked at the clonotypes that were recovered and compared them across the different approaches. And so we saw that we couldn't find as many cells with tetramer-based approaches. The number of clonotypes with frequency greater than 5 for tetramer-based approaches was smaller. And all of those were discovered with the nanovial-based approach. And then there was some overlap between the nanovial-based approach and CD137. We then look at specific comparisons between the tetramer TCRs and the nanovial TCRs, and then looking at when we re-express them in a end-fat GFP JERCAT line and expose those cells to antigen-presenting cells, what you see is that some of the really highly present clonotypes that tetramer and nanovial both discovered, they were functional. However, there was a number of other clonotypes that the tetramer approach could not find, that the nanovial approach did find that were also functional, as well as because we barcoded different nanovials with different peptides, we could also find EBV specific cells, TCRs as well, that were functional in the nanovial-based approach. We also compared with the CD137-based approach with the nanovial-based approach in CD137. We found many functional TCRs, some that we only found with the nanovial, a few that we only found with CD137. We then went and re-expressed these same TCRs in primary T cells, and we found also that the majority of these were functional in primary T cells. Again, some that we find with nanovials, some that we find with CD137 and some that we find with both. But only in the nanovials we're able to also deconvolve and understand the matching cognate peptide MHC. And we're 100% accurate in calling which peptide MHC the TCR was binding to. Okay, so we wanted to go on and apply this to finding really rare TCRs from that are cancer specific. And so we focused on prostate cancer specific TCRs. And so this PAP22 peptide is a known kind of peptide that's being targeted. These TCR156 were previously known TCRs that target this PAP22. This just confirmed that we could activate the engineered T cells that had TCR157 of 156 against PAP22. And in this case, we're using a slightly different output. Instead of interferon gamma, we're looking at granzyme B to really focus on cytotoxic cells. So after we did this proof of concept, we then did a full screen on patient sample, CD3, CD8 positive cells, granzyme B positive cells, and then on to sequencing. We also looked at granzyme B negative cells. So we compared. One thing that we did differently in this workflow is we had an oligo barcode that barcoded the granzyme B secretion level. So that allowed us to link the granzyme B barcode level to the gene expression and the TCR sequence. So as expected, the granzyme B negative cells had low granzyme B barcode. The granzyme B positive cells had much higher granzyme B barcode. And then we thought, can we use this granzyme B barcode as a way to prioritize cells that might have better TCRs, right? So if there's more granzyme B secretion, maybe that TCR is a better one. So we then we had high granzyme B, medium granzyme B, and low granzyme B. We binned them, and then we looked at in the follow-up experiment for the high granzyme B, medium granzyme B, and low granzyme B populations, which fraction of those TCRs, when we re-expressed them, were functional. So what we found is that we found more functional TCRs from high granzyme B, one that was specific. Here we did 10 different HLA-A2-1 restricted peptide MHC labeled barcode sets. So SCAMP3, MEF6, we found that were in the high granzyme B ones, and one from SCAMP3 in the medium. This is a small data set. These are only 3 TCRs that we found. Of course, previous work we had, the team had looked at 400 TCRs to find 8. So we were excited. In one patient, we found 3. But the functional TCR recovery rate for granzyme B high was around 30% or higher. And then it was slightly lower for granzyme B medium, and then granzyme B low was basically, these were not functional. So this was really exciting to think that we could start to prioritize the T cells, the TCRs the sequences that we're recovering for the ones that are more likely to be functional in that first functional screening step, right? So we're not doing downstream screening after re-expression. This is in the first functional, we call function first screen. Okay, I want to wrap up very quickly. Kind of just a little teaser on what's possible next is we're also looking at multiplexing the secretion assay. So here you're seeing TNF alpha and interferon gamma, and so here are cells that are positive for both interferon gamma and TNF alpha. We can gate those cells. So there are all sorts of questions arise, like, if cells are secreting multiple cytokines, do the TCRs we discover, are they going to be, are they going to be more functional? Are they going to have different types of functions for like regulatory T cells versus other T cells? So we think this is a really exciting direction to add more function-based screening in the first steps of screening. Finally, another kind of little teaser. We've been applying this approach also to unconventional TCRs that target MHC-like molecules. So here, this metabolite that's presented by MR1, which is an MHC-like molecule. And so, as you can imagine, cancer cells that are metabolically active might be producing more of these types of metabolites. And so we could start to target them in using TCRs that are targeting, these types of unconventional, these MHC-like molecules. and it's also not HLA restricted, so it can be applied to many more types of patients. So exciting, we can see that there's a dose response here, that we see more binding as the MR1 level increases up to some point. And that when these mate cells bind to MR1 on the nanovials, they're activated, they secrete interferon gamma, and we see this beautiful secretion signal that we don't see on the CD45 coded nanovials. So acting as a kind of artificial antigen presenting cell again on the nanovial, and we can sort out cells that are specific to this MR1 MHC-like molecule. So just to kind of summarize some of this work here, previous approaches for TCR discovery, you either had activation marker only, or you had affinity-based sorting with like tetramers. Our nanovials allow you to do both, based on both affinity and effector function, secretion in this case. And we can also barcode so that you can do multiple different peptide MHC screens all in a single workflow. And just to conclude the larger presentation, we developed really a kind of general antigen presenting nanovials that can be used both for TCR screening or antibody secreting cells to screen based off of their function, based off of affinity, based off of secretion. And this allows us to have expanded libraries of monoclonal antibodies and TCRs that are functionally annotated. So that the sequence is annotated with functional information, allow us to rank which ones we should re-express and which ones we should look at as more promising in our first kind of downstream follow-up assays. And then finally, exciting directions to look at polyfunctionality and characterize more aspects of the function of T cells in these workflows. So just to summarize, we think that we're scaling this type of biological research 1,000-fold. You think about multi-well plates, these have volumes about 1 to 10 microliters. Our nanovials have volumes that are 1,000-fold smaller, less than a nanoliter, and we can look at thousands of more compartments. And so that's beautiful to characterize function of individual cells. And I think a key aspect is you can sort and input these into other single cell instruments that are already present in labs. And that accelerates, hopefully, the adoption of these really helpful technologies, we think. So with that, I'd love to acknowledge all of the folks that worked on these projects and thank you for your time, and happy to take some questions.
Thank you, Dr. DiCarlo. It was a wonderful talk. Excellent, very cutting edge technologies there. We have time for just a couple of questions, so I'm going to go ahead and jump right into those from the audience. Lily Wong has several good questions here. The first is, she's asking if the hydrogel affects like FACS analysis, such as the scattering profiles and fluorescence signals of the stain cells.
Yeah, that's a great question. So the hydrogel is transparent, so it's mostly water, but it does scatter to some extent. So it has a slightly different scatter profile than a cell. And when you have a cell on the nanovial, that has an even different scatter profile. So actually, we gate on that combined scatter profile. In terms of fluorescence, the hydrogels, depending on the formulation and what lasers and things you're using, they're generally low autofluorescence, although in some wavelengths they can have higher autofluorescence, and so we avoid using colors in those wavelengths.
Sounds great. And Lily has another good question here. She's asking if it's possible that more than one cell can, I guess, enter into the nanovial, considering that the nanovial dimension is around 30 to 50 microns and T cells are around 6 to 7 microns in diameter.
Yep, that's a great question too. I didn't really talk through how we get only single cells in nanovials. And the way that we do that is we do Poisson loading. So we do limiting dilution, like you would do limiting dilution into a well plate. We do limiting dilution into nanovials. So we have excess nanovials, we have less cells. And so that means on average, we're getting mostly zeros. We get maybe 10 to 20% have single cells, maybe a few that have multiple cells. And so actually you can even see this in this picture here. So mostly single cells, but maybe that's 2 cells, right? So that might be 2 cells. So So yeah, so definitely we can control that through the ratio of cells to nanovials.
Great. Question from Mariko Nakano. Mariko is from Washington State, and Mariko is asking, what's the rate of cell binding, and is there any known interference for actually capturing the cells?
Yeah, that's a great question, and it depends a lot on the cell type and the types of, the adhesive moiety, and the amount of cells to nanovials, right? If you imagine you have more nanovials than cells, then the likelihood that you capture all the cells you input into the system is higher. If it's less nanovials, then you get a lower efficiency. But on average, these will range from 20 to 60% of the cells you input will be put into nanovials, depending on all these factors.
Great. I think we have time for one more question. For those that we didn't get to, we will email those over and get y'all answers from Dr. DiCarlo. The last one is from Karen Cathuria. Karen is asking, when you functionalize the nanovial, can you do it in a pooled method, or would you have individual reactions for each specificity and then combine them?
Yeah, so generally we do it in a pooled method. So we functionalize with all of the capture antibodies or antigens, and all of the secretion capture antibodies, all in one pool. If you have different barcodes that you want to put on different nanovials, or different antigens you want to put on different nanovials, then you have to do those separately, and then we mix them together.
Great. Thank you. I know we have a hard stop at the top of the hour, so I just want to thank all of the attendees. Number one, we had a great turnout today. We appreciate your time. Just very briefly, I want to say we had, of course, folks from all over the USA, also India, Nigeria, Romania, the UK, Kenya, Pakistan, Italy, Ethiopia, Peru, and Germany. And we really appreciate your presence. A wonderful attendance today. Certainly, Dr. DiCarlo, we really appreciate you taking the time to present your cutting-edge technologies. Congrats to you and your team on these advancements. To me, it looks revolutionary and it's breakthrough. And we look forward to many TCRs being discovered, and I guess other cell types being functionally discovered and isolated as well. So kudos to you and your team. And then finally, I would like to thank Max Bleckman, who's my colleague and good friend at Sino, for his IT work and flawlessly, once again, executing another Biotalk Tuesday. Max, thank you for that.
Thank you so much, Rob. It's been a pleasure. Thanks, everyone.
And with that, I guess we'll sign off and wish everybody a good afternoon or good evening or good early morning to you. Bye-bye.